Hello, welcome to another edition of the CNA's Opus. My name is Rich. I'm a certified nursing assistant, and what I want to go over today is the PI3 AKT mTOR pathway. And I apologize because I was going to go over this before, but I didn't really go that much in depth into it at all. I don't really have much time to make these videos. I don't have much time to make these videos. So I'm going to get out as much as I can, the best way that I can. So anyway, that being said, let's get started here. So this is myo-inositol, and if you watched my last video, that's the optical isomer of myo-inositol. So you could re-look that up if if you'd like, you could re-look that up on the optical isomer of myo-inositol. This here is the glycerol of the phospholipid bilayer right there, and that's your two carboxylic acid chains or fatty acids. So your two fatty acids, and there's your phosphate group right there. So right here, this would be phosphatidylainositol 4,5 biphosphate. I'm sorry, because there's two phosphate groups right here. So that's what you're starting out with. This is what is usually found in the phospholipid bilayer of the cell membrane. This is what usually is there. But once the cell wants to divide, you're going to get signals. So let's just say for the sake of argument, that you have a receptor tyrosine kinase, and the one in particular that I chose right here is vascular endothelial growth factor receptor 2, which is made up of immunoglobulin domains in its ligand binding domain right here. And they bind a little bit differently than a lot of the receptor tyrosine kinases, because it's actually the ligand, which is vascular endothelial growth factor A, that will hold these two together. You see how these are held together by the ligand. The ligand doesn't bind at a lot of them. The ligand, like epidermal growth factor, the ligand will bind at the top here, and a ligand will bind at the top of this one, and that'll cause a conformational change right here, and the two ligands will connect with one another and interact with one another. So anyway, these two dimerize together via the ligand of vascular endothelial growth factor A onto vascular endothelial growth factor receptor 2. Here is the single membrane spanning alpha helix, and down here, vascular endothelial growth factor receptor 2 is unlike other receptor tyrosine kinases, which have one domain in its kinase domain right here. It has two, okay, and in between it has a kinase domain insert. So there's a little chain right there, but it's not completely useless. It's not completely useless because it has tyrosine residues on it that can be phosphorylated. During the autophosphorylation process, there's tyrosine residues on here that can be phosphorylated. So this is not completely useless. Over here on the PI3 AKT mTOR pathway, one of the tumor suppressant protein is the phosphatase intensin homolog, which is often denoted as P10. And what that's going to do is, down here is you have, again, phosphatidylainositol 4,5 biphosphate right here. What this is going to do is, when there's a phosphate group added onto the third alcohol group here, this P10 right here will clip it off to revert it back to phosphatidylainositol 4,5 biphosphate. So I'm going to show you that in a second, but I think I got everything on this page that I wanted to. Again, you can relook this over. That's the glycerol molecule. That's the 2 fatty acids. That's a phosphate group. And that's myo-inositol. And we went over the optical isomer of myo-inositol in the last video. And I don't have much time, so I'm going to continue on here. And I hope you got something out of that page. Continuing on, this is some of the nomenclature that you'd want to know right here is the single letter amino acid code for tyrosine is Y. So I put little Y's on here on the vascular endothelial growth factor receptor 2. And these little Y's right here, once they dimerize, once both of them come together due to the ligand here, which is vascular endothelial growth factor receptor, vascular endothelial growth factor A, which actually is one gene that has 8 different splice variants, but they're all homodimers, meaning they're all the same protein, even though there's 8 different splice variants. And down here, see the ligand vascular endothelial growth factor A, and that's the ligand that we're dealing with, and then the single the single spanning membrane alpha helix, and then the autophosphorylation process, which I'm not going to get entirely into, which I actually put in my very first video. So if you want to go back to that, but I'm going to go back over that again. This one's a little bit different because when this ligand binds here and these two dimerize together right here, this kinase insert domain will flip up. There's a conformational change where it'll flip up, and these two in the kinase domain region will dimerize now. See how here before, if you looked at this one right here, they're separated. But when this comes in, when this ligand comes in and they come together here and they're activated, this kinase domain insert will loop up, and these two will dimerize. And then the autophosphorylation process will occur, and it'll become active now. And remember, these are immunoglobulin domains that are found within the entire ligand binding domain of vascular endothelial growth factor receptor 2. That's not the case for all receptor tyrosine kinases, because vascular endothelial growth factor receptor 2 is a receptor tyrosine kinase. Okay, so moving down here, I'm sorry if I'm going fast. I don't have a lot of time on this, but anyway, I'm wasting time by telling you that. Over here, this again is phosphatase intensin homolog. And the phosphatase are these enzymes that will take off phosphate groups. So this is a tumor suppressant protein right here that's going to take off phosphate groups. Okay, so keep that in mind. So this right here, when this gets autophosphorylated right here, right, it's going to recruit P85, which is the regulatory subunit, and P110 alpha. And let me just flip the page to tell you what that is. That is phosphatidylainositol 3 kinase. And 3 is the key there. 3 is the key there because it's going to add the phosphate group onto the third alcohol group right here. But it's made up of 2 portions, P85 and P110 alpha or beta. That's in all human cells except for white blood cells. So in all human cells, you're going to have this catalytic and regulatory portion in every single cell except white blood cells. So anyway, this is going to turn it into, it's going to add a phosphate group onto the third phosphatidylainositol kinase 3 kinase. phosphatidylinositol 3 kinase. 3 is the key. It's going to add a phosphate group onto the third alcohol here. Right here. You see this? Right here. That catalytic portion right there. Now, this is a transient signal because P10, which is a phosphatase, it's going to clip that off. The third right here. So this right here, is going to clip this off. Boom. Boom. So it's back to phosphatidylinositol 4,5 biphosphate. And a lot of people abbreviate it, but I like to use the full words just for a better understanding of it instead of just abbreviating it. So that clips this off and this off. And that's why that's a transient signal. But if you get 2 of them here, if you get 2 of the phosphatidylinositol 3, 4, 5 triphosphates at the same time, and it's there long enough before P10 gets to clip these off, right? It's there long enough, then it's going to recruit 2 proteins. And the 2 proteins that it's going to recruit is phosphoinositide dependent kinase 1. Now, just starting off with that one, this phosphoinositide dependent kinase 1 gets activated immediately, right away. Right when it hits it, it's a serine threonine kinase, which means it adds a phosphate group onto either a serine or a threonine kinase, right? And it binds, everything has a protein domain that'll interact with some substrate. It'll have a higher affinity to some substrate right here, okay? And the one that phosphoinositide 3,4,5 triphosphate is going to interact with is a pH domain. And pH domain is going to stand for plextrin homology. Plextrin homology. And plextrin homology is a consistent homologies. I'm going to get, I'll just briefly go over really quickly. These are consistent, conserved protein domains. The polypeptide chain is the same, right? It can be even folded the same way found in other proteins. So plextrin homology is a protein domain that's found in other proteins. It's a conserved polypeptide chain. Same amino acid sequence. Folded, maybe possibly even in the same fashion. It's conserved. So when it's found in different proteins, the first one that it's found in, that's going to be the one that's going to keep the name. So anyway, moving on from that, because that can get a little bit confusing. But this is plextrin homology, which has a high affinity to the phosphoinositide 3,4,5 triphosphate. So that is going to phosphorylate a threonine onto another protein that was recruited here via plextrin homology. And that's going to bind, and that's protein kinase B, or the older name is AKT, if you could see that right there. So this one right here is going to phosphorylate a threonine at position 308. Okay, so this phosphorylates a threonine at position 308, but that's not enough to activate protein kinase B. Protein kinase B isn't activated when it just reaches the phospholipid bilayer and links onto phosphatidylinositol 3,4,5 triphosphate. It needs to be activated by other things. So this just kind of holds it there, right? then phosphorodienosytide dependent kinase 1 is going to add a phosphate group onto threonine at position 308. Now, there's a serine over here that gets phosphorylated at position 473. Okay, so the amino acid in the sequence here, if you count it up at position 473, a serine residue is going to get phosphorylated there. And it's believed that this is phosphorylated there by the mammalian target of rapamycin complex 2, or mTOR 2. But that's the direction that the literature is pointing to, but it's still kind of iffy. All that we know is that the threonine here is phosphorylated at position 308, and the serine here is phosphorylated at position 473. And what that is going to do then is that's going to get it to drop down or cleave it off the phospholipid bilayer, and it's going to drop down into the cytoplasm of the cell. But we're not going to even go there yet. We're not even going to go there yet. One thing is that we're still at the top of the chain here. So again, we have of the PI3 kinase, we have the regulatory portion, which is P85, and then we have the catalytic portion, which is P110 alpha or P110 beta. If you don't have the regulatory portion, P110 alpha or beta will completely go out of whack and just hit the phospholipid bilayer and convert phosphatidylinositol 4,5 biphosphate into phosphatidylinositol 3,4,5 triphosphate. If that regulatory portion isn't there, that's kind of to inhibit it, to regulate it. So it'll only get activated when it's at certain sites. And in this particular one, it's at a receptor tyrosine kinase. If you don't have that regulatory portion right there, this catalytic portion will be active anywhere. It'll just hit the phospholipid bilayer and make that conversion from, I'll just abbreviate it down right now, to PIP2 to PIP3. It'll just simply convert it without that regulatory portion. That regulatory portion has to take it. It keeps it from just hitting the cell membrane wall and making that conversion, number one. And number two, the P85 domain, the P85 regulatory portion contains the Sark homology domains, which are the domains that bind to phosphotyrosine residues. So it'll only get activated at certain sites. So it'll regulate the cell cycle in that fashion. And I just want to go on just to say one more thing before I end this video, is that the PI3 AKT mTOR pathway is vital for translation, which means it's going to convert certain vital proteins like cyclin into proteins. So this is the pathway that's required for messenger RNA to be translated into proteins. The other pathways like the RAS, RAF, MEK, ERK, or Wnt beta-catenin pathways are for transcription. Those are downstream. Those are downstream for making the messenger RNA. Those are downstream for making the messenger RNA. PI3 AKT mTOR pathway is for translating that messenger RNA into proteins, and particular ones that are of vital importance for pushing the cell cycle forward like cyclins. So if you turn off the PI3 AKT mTOR pathway and you get over the insulin signaling problem, that would have a huge effect on cancer research. One thing I want to mention about cancer research here is that this right here, phosphatase and intensin homolog, is a tumor suppressant protein. So loss of function of this right here is bad news. Is bad news because if this is activated by receptor tyrosine kinase and that goes to phosphatidylinositol 3,4,5 triphosphate, and it has to be 2 of them, but there's no P10 there to clip this back off and convert it back down, then these will get recruited, and that pathway is going to remain active and just keep pushing and pushing the cell cycle forward. So loss of P10 here is not a good thing. So Rich, CNA, see you later.